In this video, we will continue with the photoprotection section. We will focus on long-term mechanisms like chromatic acclimation, state transitions, and chloroplast movement. In cyanobacteria, like in plant, state transitions is defined as the redistribution of the photosynthetic apparatus in order to balance the energy arriving to photosystem 1 or to photosystem 2. This change is triggered by the redox state of the plastoquinone pool. Therefore, in state 2, the energy is arriving mostly to photosystem 1. But if photosystem 1 is selectively excited by, for example, blue light, this induces the oxidation of the plastoquinone pool and the transition to state 1, where the photosystem 2 is mostly excited. In this state, selective excitation of photosystem 2 by using orange light that is mostly absorbed by the phycobilisomes that are the main antenna of photosystem 2, induce the transitions, the reduction of the plastoquinone pool and the transition to state 2. The molecular mechanism that take place during state transitions is still unknown and controversial. Based on different experimental observations, 4 models have been proposed. In the first model, it states that the only change during a state transition is the movement of the phycobilisome from one photosystem to the other one. In the second model, in state 1, the energy is mostly arriving to photosystem 2 through the phycobilisomes. But in state 2, less energy is arriving to photosystem 2 due to the detachment of the phycobilisomes. In the spillover model, as in the previous model, in state 1, the energy is arriving to photosystem 2 through the phycobilisomes, but in state 2, the energy is mostly arriving to photosystem 1 through a direct energy transfer from photosystem 2 to photosystem 1. This process is called spillover. In the last model, it states that in state 2, less energy is arriving to the photosystem 2 due to a dissipation of the energy at the level of this photosystem. Another long-term mechanism is the chromatic acclimation. Some cyanobacteria change the composition of the pigmentation of the phycobilisomes in response to the ambient light colors. This change is important because in oceans or lakes where usually you can find cyanobacteria, the light of different color penetrates the water in different ways. In the surface, red light is more present, and deeper in the water, blue light or green light is more present. These changes are triggered by photoreceptors that sense specific colors and induce differential gene expressions. The change in the phycobilisomes are in the phycocyanin that is absorbing red light or in the phycoerythrin that is absorbing green light. According to the chromatic acclimation, there are 3 kinds of cyanobacteria. Group 1 cannot change the levels of phycocyanin or phycoerythrin according to the light colors of the ambient. Group 2 regulate the levels of phycoerythrin. As is shown in the schema, in green light, the levels of phycoerythrins are increased. Group 3 regulates phycoerythrin in the same way that Group 2, but additionally it regulates the phycocyanin in response to red light.
State transitions also exist in higher plants and green algae. As we've seen in previous videos, this model has already been explained very well. In short, instead of the phycobilisomes moving, we have the LHC2 moving, meaning that in a system where photosystem 2 is getting a little bit more light, we actually see that the LHC2s move from photosystem 2 to photosystem 1, or the other way around. Now, this process has been shown to be more important in green algae, and it has been shown that in plants it's not so important. Another important mechanism in plants is the chloroplast movement. We don't see this process in algae or in cyanobacteria. This is due to the fact that plants have multiple chloroplasts inside their cells, and they try to adjust this according to the needs at the moment. So if we look at a cell, and if we look in normal or in low light conditions, we see that all the chloroplasts are orientated on the top of the cell in order to get the most surface area. Now we can also see that in the leaf, and when we look at this leaf under the microscope, we see that all the chloroplasts are in a way to increase the surface area. What we see is that actually all the light that comes on that is received by these chloroplasts is also taken up. However, if we move to a high light system, what happens is that the chloroplasts move to the side of the cell, and this means that we get a smaller area of chloroplast, and they tend to be to stick a little bit under each other. Now, what will happen is that some of the light will be absorbed, but a lot of the light will still pass through the leaves. And we can also very clearly see this. So this is a leaf that we have dark adapted first in order to, so basically these are two are the same leaves. And what we did was we put a little slit here in the blank. You can see the slit which has been illuminated, and the rest of the leaf has not been illuminated. And you can see that in the illuminated part we can almost see through the leaf, and that is because the chloroplasts move to the side, while in the rest of the leaves the chloroplasts are still in the original way. Now, as you can see here in the schematics, not all of the light will be absorbed, and part of the light will be transmitted. To summarize, there are several different long-term photoprotection mechanisms depending if we are looking at algae, cyanobacteria, or higher plants. So we looked at state transitions in cyanobacteria and also in plants and green algae. We looked at the chromatic acclimation in cyanobacteria, and we looked at the chloroplast movement that we find in plants.